It's actually 1.1 ZZ, just one. And every time I start with, and where did I leave off? As I was saying. So I'm gonna avoid the unit. No, this unit's big. Though you can already predict what the second half of it is. We said this unit's about metabolic processes, and we talked about metabolism in general, and thermodynamics and energy, and we talked about the sort of two types of pathways we could have. We talked about an anabolic pathway and about catabolic pathway. What we've learned about here is just one pathway in our cells. What type of pathway, and specifically a pathway that is anabolic or catabolic? Catabolic, right? We're not building something up here, we're breaking something down. And catabolic pathways can have numerous different purposes. We've focused on what portion of what we're getting out of. We're dealing with something dietary, right? Something that we take in as nutrients. In this case, glucose is the thing we've chosen to focus on because it's the most common energy source in the cell. And what we're getting out of it is energy. That's what we're processing it for here. But that's not the only reason we break things apart. It's a big one. Most of the nutrients we take in for energy, but it's not the only thing. For example, and this is something vegetarians need to be mindful of, even if you get all of your energy from plant sources and whatever, what's something you need to be mindful that you're taking sufficient amounts of in? Proteins. Why? If I'm getting my energy elsewhere. Yeah. Five proteins. First of all, it's 8 or 9, depending on where you look, but not the proteins, but the what's the name of that molecule? Amino acids. What are amino acids?
They're an amino and an acid stuck together.
That's true. With carbon in the middle, yeah, and they've got this little R group that can be different. There's 20 different types that occur in our cells, and 9 of them we define as essential, or 8, depending on what source you look at. What's that mean that they're essential? Yeah, our cells don't produce them, but we need them, which is interesting. So, like, you cannot survive without consuming the proteins of other living things. Your body simply can't do it. It is designed to bring that in, yeah.
Now I'm curious how, like, herbivores get their proteins because they eat plants.
Yeah, so the answer is the actiony parts of plant cells are protein, just like the actiony part of animals are proteins. The difference is, what's the big actiony things that animals do that requires a ton of actiony stuff that plants just don't do at all? Why do we have so much more protein than does a plant? Because again, all the moving bits of a cell, all the stuff that does stuff, all the enzymes, all that jazz, that's all protein. All the bits inside your cell that do stuff, some of it's mRNA, but the overwhelming bulk of it is protein. So why does a plant have so much less protein than we do? Because to be clear, is there protein inside of a celery stalk? Yeah, because they're cells, right? But there's not much. What is the big kind of killer app that animals have that other cells don't, that results in us having super high protein content? Just think of me. First thing people notice about me. Tough. You can admit it. It's okay. I'm intimidating. I get it. It's fine. You don't have to hide from it. I'm not going to hurt you. Yeah, I'm tough. I got muscles, right? Animals are the multicellular organism that move around. And how do they move around? Well, we actually have these special built proteins that slide against each other called actin and myosin. If we get around to it in this course, we'll learn about that structure. If you don't, you'll learn it if you take first year human anatomy or human physiology. In fact, you might learn about actin and myosin if you're taking kinesiology. I hope you would. That'd be something that I'd teach in that. Yeah. So the reason you get so much protein from animal food sources is because we eat the mussels, and the mussels are just densely packed with protein, but they're not the only thing. I just wanted to show you, by the way, really quickly that we have energy pathways for proteins as well, right? Oh, that's literally the world's smallest diagram. Right? So if we have, when we eat food, it gets digested in our mouths and our stomachs. It enters the small intestine where absorption happens, right? But if you look here, there's an entirely different process by which proteins enter into the metabolic pathways from which we get energy, right? So you can see it has its own cycle that is different from the citric acid cycle, different from glycolysis, and different from the citric acid cycle, right? This is where, sugars get processed and different food sources enter at different places. Like if you look here, triglycerides and fatty acids aren't going to undergo glycolysis, but they can actually get processed into acetyl coenzyme A. And of course, where can acetyl enzyme CoA then go? Into the matrix of the mitochondria and enter Krebs cycle. Does that make sense? Amino acids, though, not all of them, but some amino acids interact actually at the Krebs cycle level. They sort of come in partway through the Krebs cycle. Does that make sense? Which obviously means we're not getting the same bulk of energy from them that we would other amino acids can actually be converted to pyruvate. And of course, we know things that can be converted to pyruvate can then be converted into acetyl coenzyme A, which can then enter the Krebs cycle. So every amino acid actually gets processed differently. Does that make sense? Fatty acids get taken into coenzyme A. So all of our dietary sources can enter this. What is the biggest, longest, most uninterrupted chain sort of that we look at? We look at processing glucose. You should be able to predict what the next half of the lesson is. If we did the big catabolic pathway where we looked at how glucose gets metabolized down into an energy source, what's the second half of this unit going to look like? And what process goes the other way? What process produces sugars? Well, the first thing you'll note is that actually animals have systems to produce sugars too, which is that all of the enzymes involved in this process can potentially run backwards. So if your body needs to produce amino acids, it can actually do so by sort of circumventing some of this. If your body needs to produce fatty acids, it can actually do so by working glucose down into acetyl coenzyme A and then using that to produce fatty acids. Does that make sense? So the same processes that are used to break down sugars and break down fatty acids and break down amino acids can actually be used to make them under the right conditions. But the second half of this unit, we're actually going to look at photosynthesis and say, well, how did that sugar get made in the first place? So that's going to be the example that we look at of an anabolic pathway. Just to be clear, there's a lot of metabolic pathways in the cell. I used to have a poster of this hanging in my wall, but this is a rough map of the major ones, and it's by no means all of them. Oh, I don't know what that is, but yeah, so this is sort of a classic study guide where we look at just the massive bulk of metabolic pathways that we've discovered so far. And they're complicated. There's a lot going on. Oh, that's not here. So anyway, long story short, if you take like a protein and enzyme biochemistry course, you start to learn like just this massive volume of metabolic pathways. But they really, once you learn a few of them, they get a little less intimidating. But just to give a sense that all we're doing in this course is we're giving you a flavor of it. Does that make sense? We've looked at one sort of specific pathway where we start with a glucose molecule and we're trying to make ATP. And that's one pathway these things can get sent down. And that happens, what's the first step? We start with glucose. What process does it undergo? Glycolysis. Where does that happen? Cytoplasm, right? And we add the phosphate group, makes it roughly symmetrical. We use a isomerase to make it into a fructose, so into an aldose instead of a ketose. We use an aldolase to break it in half. We use another isomerase to basically make it mirror itself. And over a series of steps, we break it down into pyruvates. What's the next step? That happened in the cytoplasm. We end up with 2 pyruvates. What happens next? Yep. Beautiful. And to get from the cytoplasm as pyruvate into the matrix to Krebs cycle, the pyruvate has to be, attached to coenzyme A. Does the whole pyruvate, all 3 carbons of the pyruvate, get attached to coenzyme A? Yes, a hand motion there. Electrons are removed from that pyruvate. In the process, a carbon gets removed. It gets released as carbon dioxide. And we call that oxidation because that's losing electrons. And if the electrons that make up that bond are lost, we end up with this CO2. And then in that process, what gets reduced? Anytime something gets oxidized, electrons and energy get lost, something gets reduced. In pyruvate oxidation, what's the molecule that gets reduced? That is true, but water isn't a reduced form, actually. Water is also an oxidized form. It's a low energy state. So we have pyruvate, and I'm just going to show that as this 3-carbon molecule getting converted to a 2-carbon molecule and CO2. And you'll always see this arrow. And the arrow means that these reactions are paired or coupled. That might take some getting used to. But if you notice that whenever you see 2 arrows together, that means that the energy released by one reaction is driving the other reaction. Does that make sense? They are paired. So as pyruvate becomes acetyl coenzyme A, we could even, if we wanted to, draw the coenzyme A molecule there, but who cares? What is the energy storage? Energy gets released there, and it goes into NAD plus H plus will yield NADH, right? Does that make sense? So that takes a bit of getting used to, but you want to get in the habit of understanding these as paired reactions. We draw them sort of articulating against each other like that because for this reaction to happen, it needs to get energy, and that energy gets released by this. We say that this is oxidized. In other words, it's going to lose electrons. And we say that this then is going to be, whenever one thing is oxidized, oil, the other thing is reduced, which is just a fancy way of saying something loses electrons, loses energy, and the other thing involved, the other reaction will be one where energy is gained, which makes sense because energy can't be created or destroyed, right? So if one thing is losing energy, another place is gaining it, right? And if we have a reaction happen side by side, we can store that energy. Cool, then we go through Krebs cycle. Krebs cycle has a bunch of steps of oxidation and reduction, right? And that's what we're showing every time here. I hate about this diagram that it has these arrows kind of go in the opposite direction, though I think it's almost trying to prove the point of opposite in direction. But do you see how this arrow just comes off of this arrow? Because this CO2 is released. It's not the product of another reaction. It is the reaction of isocitrate into alpha-ketoglutarate releases a CO2. The isocitrate is being oxidized. But if the isocitrate is being oxidized, then something must be being reduced. And what's being reduced? The NAD+. I always put the H plus with the NAD+. I don't know why the H plus goes with the NADH. I think in my mind, I think that that's like NAD with an H stuck on it, which is not actually what that means. But anyway, don't make the same mistake I do. So during Krebs cycle, we charge up an NADH here, we charge up an NADH there, we charge up an NADH there. Of course, for every glucose, how many times do we go through Krebs cycle? 2, so we actually get 6 NADHs. We also charge up a GDP into GTP. That'll later go on and get converted to an ATP. And we also charge up an FAD into an FADH2. Each of these is an example of a reduction reaction. In each of those cases, these molecules are being reduced. And reduced is a weird way to say gaining energy, right? I hate that language. I really wish that wasn't the language we used. Oxidizing and reduction are possibly the 2 worst terms in science because they have nothing necessarily to do with oxygen, and reduction means gaining energy and gaining electrons. Do you see how this is bad naming? Can we all agree that's bad naming? So if you find that you get that one mixed up or confused or tripped up, the name is designed to make it counterintuitive and thus to make it not stick in your brain easily. You play with the words long enough and you use that language long enough, it'll make sense. All of these molecules that now are charged up with energy, the only one that the cell can use directly is the ATP, and we haven't made any ATP since glycolysis. So how do we make ATP out of all the rest of these? Except for the GTP. We haven't talked about it. It has its own little metabolic pathway. How do we get ATP from the NADHs and the FADH2s? You're either going to a luau or you're trying to tell me that they enter the electron transport chain. The electron transport chain is responsible for what we call oxidative phosphorylation. And the process itself, our understanding of how this works is what we call the chemiosmotic hypothesis. A lot of words there. But what does it mean to oxidatively phosphorylate something? Well, phosphorylation is how we charge up ADP, right? We charge these ones up by adding electrons, but we charge ADP up not by adding electrons actually, but just by sticking another phosphate group onto an adenosine diphosphate, ADP, to form ATP. How does that happen? Well, it happens sort of by what we call the chemiosmotic hypothesis. And the basic idea here is that, where's my, there we are. The basic idea here is that the NADH comes and it gives its electrons, 2 of them specifically, to a protein that can use the energy from this being, now if this is losing electrons, the NADH, which was reduced in a previous step and given energy, is now being oxidized, which is another way of just saying it's losing energy, right? So if it's oxidized, that means this protein complex is being, what's the opposite? Anytime something is oxidized, something else is reduced, which means this protein complex gets a big surge of energy. If it gets a big surge of energy, what does it use it to do? Pump an H plus ion. Actually pumps 4 of them, but you don't need to know that. We always just draw it as 1 because it's not the details that matter. Those electrons get passed to a protein complex called quinone, which then carries them to what we call complex 3, which is, and again, each of these is an oxidation reduction step. And as you saw in that video, there's actually a bunch of oxidation reduction centers within these proteins. The electrons get passed around like a hot potato until they get to the active site where quinone binds, and then they get passed to quinone, which has its own couple sites. And then as the electrons move through quinone, they change its shape. It's no longer favorable to bind to complex 1, but now it's favorable to bind to complex 3. When it does so, it passes the electrons. Again, they get passed around inside complex 3 like a hot potato, and all of the energy that they expend there is used to do what again? Pump H plus ions. From there, they get passed to the cytochrome C complex, which carries them over to complex 4, where again the hot potato thing happens, which again causes an H plus to get pumped. So there's 3 locations where an H plus gets pumped. Complex 4 actually is the final resting place of those electrons. They get given to oxygen, which now means that oxygen will have 2 extra negative charges, which means it's going to want to find 2 things that are positively charged. What does it find? H plus. Where does it find H plus? It's just in the water, because what's always in water? H plus. And that forms water. Now the electrons are gone. All the energy from them has been used to pump H pluses up there. This is the equivalent of pumping a whole bunch of water up into a tank on the top of the roof. Does that make sense? If you just leave it up there, it'll just stay up there. But if you give it a path down, what will it do? Come down. If you give it a path down and you put a turbine at the bottom, what will it do? It'll spin the turbine and it'll make electricity. You can capture that energy. That's literally what this is. It's a mechanical turbine, only it isn't used to make electrical energy. It is used to mechanically stick an ADP to a phosphate group forming ATP. And that's how we get energy. But for this to happen, the electrons have to be able to flow. And the electrons can only flow if they can always get passed on like a hot potato. And they can only keep flowing and get passed on like a hot potato if complex 4 can get rid of them. And how does complex 4 get rid of them? It needs to bind to oxygen. So without oxygen, this whole process breaks down. Does that make sense? That's just the way it works. It's just the way it's evolved. Which means, You take oxygen away, what happens to this whole thing? It stops. And if your cell stops producing ATP, what's going to happen? You're not going to have enough ATP. And if you don't have enough ATP, what's going to happen? On a big scale, as a human, this happens to us because if we are running low on oxygen, our body will divert it to the most important places, which for us, of course, is our heart, our diaphragm that keeps us breathing, our intercostal muscles that keep us breathing, and our brain. And every other muscle is considered kind of secondary. Every other system is considered kind of secondary. So you've probably experienced, if you've exercised really hard, like me, the experience of running out of enough oxygen for it to work, and you've probably felt your muscles kind of shut down, right? You ever push through that? Because we can. What do you experience then?
Ow.
We experience ow. Correct. And that's what today's about. Today's going to be about experiencing ow. Hopefully just learning about it. Hopefully this isn't like physically painful for you. Ruby, it's the important part now where we learn something new. All right. So today we're going to learn about what we call anaerobic respiration. So recall that the normal cellular respiration results in the production of 38 ATP molecules from a single glucose molecule. By the way, are all 38 of those available? Because at a minimum, 36 are available because at least 2 get spent in the process. But all the same, that's the rough ratio. 1 glucose is equivalent to about 38 ATP molecules in energy. But we know that this is under normal conditions. And under normal conditions, this requires oxygen at the end of the electron transport chain. This requires O2 to act as a sink for electrons I guess we'll say 4 electrons because it's 4 electrons for every O2. Well, actually, I'm going to stick with that 2, and you know what I'm going to do is that obnoxious thing is put a half on the O2. But that's okay. For the 2 electrons at the end of the electron transport chain. That's why we call it oxidative phosphorylation. Now, a cell biologist would call this oxidative phosphorylation. What would a kinesiologist call the process of getting energy in this way that requires oxygen? You could also call it aerobic respiration. In the 80s and 90s, aerobics videos were big. These were like videos that you could buy, and it would be like people working out on screen, and they would like lead you in an exercise. But the idea with aerobic exercise is that you're not straining your body to the point where it's short on oxygen. You're exercising for a good long prolonged period, but you're always maintaining sufficient oxygen levels that your muscles are never under strain. Today we're going to talk, however, about anaerobic respiration. So what is that? No oxygen. Good. So we would say under normal conditions, we need 6 O2 molecules to break down a single glucose molecule. If you go back, you can do the math and see that that works with what we've learned. Without O2, the electron transport chain shuts down. But of course, the electron transport chain, remember we said that it's not like a new NADH gets made every time we go through Krebs cycle. In Krebs cycle, do we make NADH? No, we just re-energize it. Do we make FADH2? No, we just re-energize it. So if we run out of the electron transport chain and it can't keep working, all the NADH that gets produced by Krebs cycle will eventually mean that what don't we have? The precursors. Does that make sense? If you're making all this NADH, but you're not then unmaking it, you're going to run out of NAD plus, and you're going to run out of FAD. Does that make sense? So, since the ETC oxidizes NADH back into NAD plus and FADH2 back into FAD, without it, is everyone comfortable with W/O being without? I'm just going to say this is a bit of a simplification, but we'll say runs out of NAD plus and FAD. The truth is it wouldn't run out, it would just run in low concentrations. But the upshot of that is what can't run without NAD plus and FAD available? Krebs cycle. Does that make sense? So that would mean that the Krebs cycle shuts down. Yeah. Sorry? Yeah, there's an H there, isn't there? Thank you. Now, we made a note earlier and we said glucose is not the only source of energy for the cell. And we said that other nutrients get processed in different ways. But also involve Krebs cycle and the electron transport chain. Yep. Sorry.
All the other previous notes just say it's FAD to FADH2.
Well, now you're causing me to stress out. Now I'm going to go double check. Now I feel stupid. Let's check the previous note, then we'll know. Yeah, it's just FAD. See, I had it, and then somebody told me I was wrong. And a fun fact about me, if somebody tells me I'm wrong, I assume they're correct. It's okay, listen, I respect it. And I'll get it wrong. I'm not blaming you, believe me. I'm the teacher, I get paid to know this stuff. I have a degree in this crap. At least I would if I hadn't filled out my graduation request form wrong. Anyway, that's a different discussion. Yeah, which is still this stuff. So that's the thing I want to note, is that, so even other nutrients, so all nutrients eventually require O2 for ATP production. In other words, that oxidative phosphorylation is the way that we get our ATP. Except for where? Except for where? Where's the one place that's not true? We get all of our ATP from the electron transport chain and then the ATP synthase molecule, allowing those H plus ions to flood back into the matrix of the mitochondrion, turning the stator portion of the ATP synthase molecule and mechanically forcing together ADP and phosphate to form ATP. But there's one other place we get ATP. Where is it? Glycolysis. So if you look at the next page, and I want to give you a minute to write this. So do you need a minute or can I go on? You need to, okay, yeah, take your time. I don't want to rush you. If you look at this diagram on the next page, and you don't need to know this, but I actually think it's, again, a nice little diagram to show you just the idea that different molecules enter our metabolic pathways at different points. And keep in mind, as this arrow makes clear, these arrows are doing a lot of work. Does that make sense? Like, each of these is its own metabolic pathway, right? Just like glycolysis, we know isn't actually just an arrow, it's actually 10 steps, and it involves 10 different enzymes doing a whole bunch of different stuff. Lipids being broken down into fatty acids requires a bunch of steps. And then those fatty acids eventually being converted to acetyl coenzyme A requires a bunch of steps. We're just not learning about them here. Does that make sense? This is complicated stuff. But regardless, where do they all end up? They all end up in the citric acid cycle, also known as Krebs cycle. And from there, where do they end up? Well, they end up acting through these proteins that are bound to the membrane, separating the matrix from the intermembrane space, known as the electron transport chain, or collectively referred to as electron transport and oxidative phosphorylation, right? Which is just the idea that we take all of these intermediates there, but that can only work if we have supply of oxygen, all right? So like we said before, again, without oxygen, it all shuts down. But glycolysis is anaerobic. And what does anaerobic mean? Does not require oxygen. And produces a net of what? Two ATPs. So without oxygen, this is all the energy that can be gathered. How efficient is that compared to? What's the efficiency of that? Mathletes? Well, 2 out of a normal, what would the normal amount be? Yeah, we'll call it 36 in net. That can be reduced to 1/18th, right? What would our math be there? X would be equal to 100 divided by 18. Oh, that's not true. Oh, yeah, sure it would. 100 divided by 18, which is how much? 5.5%. So that's not very good. But in the absence of oxygen, we can only get about 5.5% of the energy out of our glucose. Yeah. Ah. Which is actually going to be a big problem for us. So you're exactly right. Look at this next title. Boy, you're good. Glycolysis rather, also nets not 1 NADH, but 2. Yeah. Yes. Yeah, sorry. How's that? Now, normally you would think that getting NADH out of glycolysis would be another advantage, but of course, what's the only way the cell can use NADH? It has to turn its energy into ATP, and where does that happen? At the electron transport chain, and what does that require? Oxygen. But it gets worse. You see, your cells are designed to not constantly undergo cellular respiration. When you're sleeping, do you want to burn as much sugar as when you're running a race? It has to be regulated. How does the cell regulate when to undergo glycolysis and when not? Because remember, glycolysis kickstarts the whole thing, right? Yeah.
We have too much of the other big stuff, so like you can't.
Yeah, and as it happens, the specific inhibitor of glycolysis is NADH. So glycolysis is regulated by NADH levels. In other words, NADH is what we call an antagonistic inhibitor of glycolysis. It can bind to some of the key enzymes involved and stop them from working. So NADH acts as an inhibitor. What does it mean if something is inhibited?
Mr. Calford, please contact the office.
That's so messy. I'm really messy today. I'm sorry. Another way of saying this is when concentrations of NADH are high, glycolysis goes slower. And if they get high enough, it will actually stop. So here's the other problem. This means that without oxygen, NADH levels rise, and the cell doesn't even get those 2 oxygens, or 2 ATPs rather. The cell can't even get 2 ATPs because glycolysis will stop. This is where, if you were designing a cell from scratch, you would not make this the inhibitory method. Does that make sense? It is a really bad system. This is a bad design that we're designed to say, okay, the thing that if we run out of oxygen, we're only going to be able to get 5% of our energy from glucose. And then to say, oh, by the way, the mechanism we've designed to tell the cell when to do glycolysis and when not, in the absence of oxygen, we're going to tell it to stop doing even that. Is that a good design? It's a bad design, but it's just how it works. Because it wasn't designed, it evolved. Does that make sense? So the regulatory method by which glycolysis is controlled is normally, and you can imagine that normally for the cell, this makes a lot of sense, because if ATP isn't getting used up, then eventually ADP levels will go low, which will make the electron transport chain slow down, which then NADH levels will get really high. That will make Krebs cycles slow down, and then that is designed to also say, well, if that's happening, there's no point having glycolysis keep producing acetyl coenzyme A and shoving it into the matrix. So glycolysis has evolved to also slow down when NADH levels get high. But if you are starving of oxygen, you want to keep producing energy, right? So what do we have to get rid of? We have to get rid of the NADH. So fermentation is the process by which NADH gets processed so glycolysis can keep running in the absence of oxygen. And there's two major methods. And the first is what we call alcohol fermentation. Okay, so in alcohol fermentation, we have to figure out a way to get rid of those NADHs. And the way we do that is that we take our pyruvates and we actually do just what would happen if we were going to send it into the mitochondrion, which is we take a carbon off each one, producing what kind of group? An acetyl group. An acetyl group, when it's not bound to anything, we just call acetylaldehyde. This is the acetyl group in just its aldehyde form. In other words, it's just its own little molecule. But instead of sending it into the matrix, it's just dealt with in the cytoplasm. So this happens in the cytoplasm. So what we want to understand is that there's a problem. The problem to be addressed is get rid of NADH without using oxygen. So how's that happen? Well, the answer is that a carbon is released from each pyruvate. And that leaves us with two acetyl groups. And then the acetyl groups are processed into ethanol. What's ethanol? It's an alcohol. It's a 2-carbon alcohol, but it is what we refer to pejoratively in the vernacular just as alcohol. This is like when you drink an alcoholic beverage, which of course none of you have done because you're underage, but one day if you should, and you shouldn't because it's terrible for you. It's a carcinogen and a neural toxin and makes people do dumb things and behave in ways that we find antisocial. And if people behave that way when they weren't under the influence of alcohol, we'd think that's a terrible human being. I don't want to be around them. But anyway, enough about that. Ethanol is just a byproduct. All right, it's just a byproduct. The reason it produces ethanol is that this involves an energy gain, actually. Ethanol is a higher energy molecule than acetaldehyde. This is a reduction. And the nice thing about this being a reduction, it means the energy has to come from somewhere. And where does it get the energy? The energy to turn acetaldehyde into ethanol comes from the NADH we made during glycolysis. So the upshot of this is that by turning pyruvate into acetaldehyde into ethanol, the NADH produced by glycolysis is oxidized back down to NAD+. And this allows glycolysis to keep going. Again, this means that the cell is desperate, all right? In the lack of oxygen, it has to do this just to get 2 ATP per glucose compared to a normal rate of 36, and an efficiency of about 5.5%, drastically reduced. But of course, there's another problem. What's the new problem? Ethanol is toxic and will eventually kill the cell. So the cell can do this for a while. It will extend its life, but if this happens long enough, it will produce enough ethanol that it will die. And yeah. No, humans actually have a different. That is just good old-fashioned. Your body is depriving parts of your brain of oxygen at that point to keep more fundamental systems alive. And probably I would bet that having you get dizzy is an evolutionary advantage for your body to say, slow down. We need to get more oxygen and do less moving. So there's probably an evolutionary advantage to having you feel a little discombobulated when you're running out of oxygen. But of course, runners train themselves to push through this, right? Yeah, we don't ferment. Alcoholic fermentation takes place in some plants. It takes place in fungi. The classic thing that we use for fermentation is what? We use it in everything. No, grapes, we use them to get sugar as a source. Could be grapes, could be potatoes. But what do we actually use to ferment grapes and things like that? If you leave grapes on their own, it's true they will ferment, but they will do so very slowly. And plants, as it happens, don't ferment nicely. They produce a lot of other alcohols too. And so you don't get like a nice thing like that. That's like vinegar going bad, or wine going bad tastes like vinegar. That's the plants kind of doing their thing. We use yeast. In fact, we use a particular brand of yeast. You don't need to write this, but called, quiet you, Saccharomyces cerevisiae. And Saccharomyces cerevisiae is like the yeast we use for everything. We use it in bread making. Interestingly, in bread making, what do we want it for? In bread making, we don't make it do this for very long because you don't want your bread to taste all boozy. Though there are some breads where we leave it longer and you start to get these alcoholic, a little bit of that there. We have bacteria that will actually do this a step further where they'll break this ethanol down into acetic acid. And that's where you get sourdoughs. But the main thing we want this for actually in bread isn't the alcohol, it's the CO2. That's what makes your bread leavened. That's what makes it rise. Is we take this poor creature, this poor yeast, Saccharomyces cerevisiae, and we mix it with a bunch of flour, which is full of sugar sources for it, so it can start eating it, and it's like all happy. It's like, oh, hey, look at this. But then we make it into this big pasty dough where there's no oxygen in there. And then we go put it in an oven. So everything speeds up and it starts metabolizing like crazy, and it quickly runs out of oxygen, and in a desperate bid to survive, it starts fermenting and producing all sorts of CO2, and that puts all these nice little pockets of air in your bread, and that makes it all nice and fluffy. You murderers. All you vegetarians are like, oh, look at me, I don't eat animals. No, I just bake living things while depriving them of oxygen to get them to desperately try to survive to produce CO2, and then finish it off by baking them to death anyway. But yeah, so when we bake with Saccharomyces, we like the fact that this process produces carbon dioxide. On the other hand, when we put Saccharomyces in a vat of grape juice and then seal the top so it can't get any oxygen and leave it for months and months and months and months to die a slow, painful death by the toxins it produces along the way. Yeah. What we want out of that is the ethanol. And in the past, people drank much, 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 much more than people drink today, because interestingly, in the past, in a lot of places, alcoholic beverages were people's sort of only source of clean water, or the only reliable source of clean water, because that alcohol kills everything in it, including you, but you more slowly because you're big. and the little things like that. And of course, in modern days, we have good water filtration techniques. But if you've ever read, you should read sometime a book about like the British Navy and see what their alcohol ration for a day was. And it was like a staggering volume. And then you think, I don't know if I'd want to be at sea with a bunch of people like facing like 20-foot waves where everybody is just like pissed drunk. But that is in fact how it was. Yeah. Well, again, your body doesn't do this because our body doesn't produce ethanol. But what your body does with ethanol is the first thing it does is absorbs it into our bloodstream, and it is a small little molecule, so it crosses the blood-brain barrier, and it immediately starts giving you brain damage, which is what being drunk is. It's just brain damage. But at the same time, your body, knowing that this is poisonous, it gets processed and broken down into things that we can excrete safely. And where does that happen? The liver, or at least at other places as well, pancreatic action, kidney action, but primarily the liver. So your body has to break down ethanol into other things, which it can safely do at a certain rate. And then when you begin drinking beyond a certain level, which I mean is basically anything, your body can't keep up, right? And so it takes time for it to catch up. Yeah. Questions about that? What do? We don't do alcoholic fermentation. Yeah, so you're not going to run and get drunk. But you've probably run and gotten a cramp. What's that feel like? It feels like ow, yes. It feels like a sharp shooting pain, right? You felt that before? That sharp shooting pain you're feeling is being burnt by acid because you're being burnt by acid. So humans and some other bacteria and some other things, but we'll just focus on us for the time being. Humans undergo a similar process. Where pyruvate is processed using NADH being oxidized down to NAD plus to allow glycolysis to continue. In humans, the byproduct is not ethanol, but lactic acid. Somehow the U looked more like a V than the V. The one thing I want you to note is that, well, two things to note. One, this is what causes cramps during anaerobic exercise. Now, to be clear, it's not every cramp is not caused by lactic acid. Lots of things can cause cramps. Often it can just be muscle inflammation and things like that. But if the specific cramp you get, that sort of very specifically sharp, I don't know how to describe it, but it very much feels, it's not like a throbbing pain, it's very sharp pain, right? That you get when you're like running too hard. This is what causes cramps during anaerobic exercise. But note too that there's a little difference. Lactate, which is another word for lactic acid, just like citrate is another word for citric acid. Lactate is a 3-carbon molecule, which means what doesn't get released here? Yeah, so we actually don't produce carbon dioxide when we ferment, whereas things that undergo alcoholic fermentation do. Yeah, how's that? Can you read that?
No, yeah, your record player's kind of blocking.
Oh, I'm sorry. How's that? That's the end of my note. So we've got that.
That makes it a little better. We don't need to draw oxygen elsewhere.
Yeah, and there is a note. This is actually a one-step reaction, a rare one-step reaction. Yay. Remember, in lactic acid fermentation, it was a two-step reaction. The pyruvate got whittled down into an acetaldehyde, right? And then the acetaldehyde got broken down into ethanol. In this case, one step. Pyruvate becomes lactate, and that requires NADH to become NAD+, and that allows that to go. Unfortunately, lactate is also toxic. Ethanol, because lactate can't cross the blood-brain barrier with ease. By the way, we will make a quick note. Lactate is the same thing as lactic acid. Not as easily. It can, but it doesn't do so as readily.
I feel like we would be having a lot more splitting headaches.
Yeah, and I can't imagine that we would. I don't know the details. Again, I'm not a neurobiologist. I took 2 neurobiology classes, but famously I skipped every single lecture of one of them and just did the exam because it was only a final exam in that course. 92, baby. All right. How much of that do I remember? Sadly, none, which is why it's important to actually go to the lectures and engage with the material and not just cram and study for a test. But you know, I did all right. Can somebody remind me if the textbook section I gave you included pages 134 to 137? I think it should. If it doesn't, I will print more for you. But I do have your homework here, which the homework is I want you to read. Does it? Damn it. Okay. Pages 134 to 137 for comprehension. And I can't stress enough, if you're not doing these textbook readings when I give them to you, boy, please do. Learning how to read a textbook as a learning tool is dramatically helpful. And then I have some questions for you to think about. 9, 14, 15, and 16 on page 137. But I don't care about the answers just to these questions. And I don't care about the answers at all. I care that you understand it. So I can't stress enough that, like, don't say I'm not going to do the reading, I'm just going to should be able to read question 9 and go, oh, I know the answer to that. And if you don't, then you know what you need to do? You need to reread it, or maybe talk your way through it, or maybe write it out, or maybe call up a friend and be like, hello, friend, I just did the textbook reading of page 134 to 137. I was wondering if you want to talk your way through it with me. Something like that. Yes, sir.